This video, Chapter 6, Part 4, is really going to focus on enzymes. Enzymes, as you might remember, are proteins made in living cells, and they catalyze chemical reactions. So, I guess we need to know what a catalyst is. And a catalyst is any chemical agent that speeds up a reaction, but itself is not consumed by the reaction. So that's the general name for a catalyst. An enzyme is a really specific macromolecule that acts as a catalyst. It's usually proteins. And so here's an example of how the enzyme, the protein enzyme sucrase, which we see right here, is used to cause the hydrolysis of sucrose. So here's sucrose, which you might remember from when we started talking about chemistry earlier, organic chemistry. Sucrose, we're going to break that into its two subcomponents, glucose and fructose. And notice that glucose and fructose have the same chemical formula, even though they are different from each other. If you want to remember what it means when you have the same chemical formula but different molecules, you can go back and review your previous lectures. So we need some water here, and the water is causing these, it's going to be added onto the glucose and fructose in the presence of this enzyme sucrase. And do you remember what that's called when you use water to break down molecules? I hope you do, because we've been talking about it in this chapter as well, especially focused on ATP. So that's your enzyme. It's going to allow that chemical reaction to happen more easily. Let's take a look at what it means to have a chemical reaction happen more easily. Do you remember that picture I showed you of that person pushing a rock up a wall? Well, chemical reactions, even the ones that happen spontaneously, do have a little bit of wall or mountain or hill that they have to climb before they can go through their chemical reactions. This barrier to the chemical reaction starting is called the activation energy, or E sub A. Energy of activation is how you say that, or activation energy. So we've got to break bonds before we can reform bonds. And activation energy is often supplied by heat of the surroundings. So if it's a warm surroundings, then the energy of activation can be overcome. But sometimes that's not enough. So what we're trying to do is go from this AB complex and this CD complex, and we're going to stress the bonds between AB, and we're going to stress the bonds between C and D. That's the energy of activation, where we start stressing those bonds. And again, we can do that with heat. And then we reform these bonds between A and C and B and D. This has a delta G of less than 0, which means it is spontaneous. You can also just look at this graph and tell it's spontaneous because the free energy is higher in the reactants and lower in the products. So, how do enzymes speed up reactions? Well, we were just talking about heat, right? And heat is non-selective, meaning hot things, if it's hot, a lot of different reactions can happen. It's not selective. It's just going to make any activation energy be overcome of any chemical reaction. But we want specific reactions to happen in our bodies. And so instead of relying on heat, which, by the way, heat in general can be pretty bad for your body. Think about fevers, right? If you have a fever, your proteins become denatured, and that means they can't do their job. So we don't want to rely on heat. It's not specific, and it can really mess things up. No, we're going to rely on enzymes. So enzymes are going to be the catalyst instead of heat in a living organism. Let's take a look at how that happens. So here's in black, this is the course of the reaction without the enzyme, and we've got that hill that we got to get over, right? What enzymes do is, it's not like they affect the delta G. It's not like they affect the amount of energy that's released, right? The reactants are still there, the products are still there, we still have a negative delta G. But instead of having to have this much energy be put in, we only have to have this much energy put in. So the energy of activation is less, or we say that the enzyme lowers the energy of activation of a chemical reaction. That means that if they're lowering the delta G, they speed up a reaction that would normally not occur as fast without them. So let's take a look at enzymes and get some terms associated with enzymes so we can learn a little bit about them. First of all, enzymes are very specific for the reactions that they catalyze. Notice before we had the sucrose, which was being broken down by the enzyme sucrase, and it was going to its products, glucose and fructose, right? Sucrase only acts on sucrose. So we say that enzymes are very, very specific, and they're so specific we get to name them after the chemical reactions that they mediate. The substrate is the reactant. So in enzyme-mediated chemical reactions, instead of saying reactant, we often say substrate. And the reason for that is because that's the word that we use for something that binds to an enzyme. We say that's the substrate. Again, it's the same word, substrate, reactant. It means the same thing, but substrate implies that we've got an enzyme working on it. When the enzyme binds to its substrate, it forms something called the enzyme-substrate complex. That's pretty straightforward. Interestingly, most enzyme names end in ASE, like when we were talking about sucrase earlier. Sucrose, OSE, that's carbohydrates, right? Simple sugars, they end in OSE. If it ends in ASE, it's most likely a protein-based enzyme. So let's take a look at how that substrate binds to the enzyme and what happens next. So here's our substrate, and here's something called the active site. And the active site is the region of the enzyme where the substrate binds. If the substrate bound over here, then this would be the active site. But this is where the substrate goes, so that's our active site. Enzyme specificity happened. Remember, enzyme specificity, that's where a specific enzyme only binds to a specific substrate. This happens because of the shape. So here's the specific shape of the active site, and only this substrate is going to fit into that shape of the active site. Again, we call that enzyme specificity. Enzymes, when they get their substrate bound to them, they change their shape. It looks like this enzyme is kind of hugging the substrate. And when it changes its shape and binds onto it, alters its shape, we now call this the enzyme substrate complex. The substrate kind of forces the enzyme to change its shape in a process that we call induced fit. So that's induced fit, the enzyme, and it's the change of shape in the enzyme, and it's caused by that substrate. So the substrate is held to an enzyme's active site by weak interactions, like hydrogen bonds. The active site then lowers the energy of activation by converting, and then it converts the substrate into products. But look what happens here. Here's our enzyme, and it changes its shape. When it changes its shape, it forces those two parts of the substrate to kind of stretch and break a little bit, and now we have our products that are in their separate molecules. So that stretching and that breaking, that's the lowering of the energy of activation. Let's take a closer look at this, putting it all together. The active site is where the substrates are going to join, and here we have our substrates right here, the red and the green substrates. they're going to fit into the active site, which is going to cause that change in the active site. Here we've got some weak interactions going on. Hydrogen bonds, could be ionic bonds, something along those lines. The formation, that change in the shape of that enzyme, that's what's lowering that energy of activation. That's what's speeding up the reaction. The substrates are then converted to the products in that active site, and then the enzyme releases those products, and now it's ready to, it's like available again for some more substrates to join in. So the enzyme is not used up in this process, and the substrate is converted into product. Remember, substrate and reactant, same thing. The active site can lower the activation energy and speed up the reaction in any one of 4 ways. And I talked to you about one of them already. But one is it orients the substrates correctly. It lines them up. It makes sure that they're ready to exchange atoms. Straining the substrate bonds, that's the one I was talking about, where it bends and stretches a little bit, straining substrate bonds. It can also provide a good microenvironment for the substrate to be ready to change. And it can then cause covalent bonding to the substrate. So lots of ways that we get the enzyme's active site being important in this chemical reaction. Other things that are important in enzyme chemical reactions is how much substrate there is compared to how much enzyme there is. So the rate of the enzyme catalysis, right, that's chemical reaction, right, can be sped up by increasing the amount of substrate, or the substrate concentration. So here we have a substrate at 2.5%. The substrates are the little pinkish red ones, and the enzymes are the little C-shaped ones that are in blue. So enzyme, 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 and this one right here, that's the substrate. As you increase the substrate, as you increase the substrate concentration, you're increasing the rate at which the chemical reactions form. As you add more and more substrate, it seems like it would just go faster and faster. But when all the enzyme molecules are bonded with the substrate, we say that the enzyme is saturated. When enzymes are saturated, the reaction speed can only be increased by adding more enzymes. So if your substrate is at 80% and you that's more than half of the solution, right? So there's more substrate than enzyme. It's not like eventually those won't get converted by the enzymes. It's just saying you got your balance a little off, and we can't speed it up unless we have more enzymes. Remember, we already talked a little bit about how proteins are affected by their environment. So an enzyme, because enzymes tend to be proteins, enzymes can also be affected by their environment, the same kinds of things. So temperature and pH, those are kind of things that can cause the enzyme to denature. And if an enzyme denatures, it can no longer perform its function. If we take a look at the effect of temperature and pH on enzymes in a graph, we can see some interesting things, like not all enzymes are the same. So here in this first graph, we have a thermophilic cyanobacteria. That's fancy. Thermophilic just means likes hot environments. Thermophilic loves heat. And cyanobacteria, cyano, that's a kind of color of green, so it's a green bacteria. In other words, it's a photosynthetic bacteria. Now, if you look at those thermophilic bacteria, you can see that they really do their highest amount of work, their speediest amount of work at a really high temperature. This is in Celsius. So remember, boiling hot water is right here. So we're talking about in the 75 degrees Celsius range. Now for humans, our body temperature is about 37 degrees C, and the enzyme shown by this black curve is an enzyme found in your body, a typical enzyme found in your body, and its best rate is right around body temperature. So you can't really say that there's one ideal temperature or one ideal pH, and we'll talk about the pH one in a second, but for all enzymes, they do all specialize. For example, here for pH, this is pepsin, which is a stomach enzyme. And I don't know if you remember, but stomachs have a very low pH, meaning they're highly acidic. So this one does really well at a pH of 2, very acidic. Trypsin, on the other hand, is an enzyme found in your intestines, and its optimal pH is at 8. That's a little on the basic side of the pH scale. Remember that 7 is neutral on the pH scale. So enzymes do best at specific temperatures and pH depending on what enzyme you're talking about. Something else that really can affect enzymes is something called cofactors and coenzymes. We'll be talking about this quite a bit when we get into later chapters. I grabbed this picture off the internet. It is very useful. It looks maybe like it contradicts something that I'm going to show you in a minute, but it doesn't. So let's look at what cofactors and coenzymes are and see how they work. So cofactors are non-protein molecules that help enzymes carry out processes that are difficult. So cofactors can be inorganic, such as like metal when they're ions, or they can also be organic. If it's organic, we give it a different name. If it's organic, we call it a coenzyme. So cofactors is a general term for a non-protein molecule that helps the enzyme and can be inorganic or organic. Coenzymes are always organic. So take a look here at this cofactor or coenzyme, and here's that active site, right? This cofactor or coenzyme fits inside that active site, binds to the enzyme, and it kind of changes the shape of that enzyme so that this flat bit of this substrate can fit very nicely in the flat bit of the enzyme. And that's what is happening here with the cofactor or coenzymes. Vitamins, they're vital amines, right? But they actually act as coenzymes. They can also act as raw materials from which coenzymes are made. So vitamins are pretty important for staying alive because they help with our enzymes. Now, we can also have things inhibit enzymes, and this sounds like a bad thing, but it's not necessarily bad, as we'll learn. So enzyme inhibitors can modify or regulate enzymes. We can have competitive inhibitors or non-competitive inhibitors. Competitive inhibitors bind to the active site. So here's your enzyme, here's your active site, there's your substrate, and normally that would fit just in there really, really nicely, and the chemical reaction would happen. When you've got a competitive inhibitor, that little inhibitor binds to the active site, and that doesn't allow our substrate to bind on. So the substrate can't bind onto that active site, and that means that the chemical reaction is not going to happen. Non-competitive inhibitors are very interesting because they also change the active site, but not at the active site. So this non-competitive inhibitor down here is binding to some other region of the enzyme, which causes the active site to change shape. And when the active site changes shape, it's not going to bind onto the substrate. So it's going to become less effective, or it might become completely ineffective by that non-competitive inhibitor. There are some enzyme inhibitors that are reversible. They bind to enzymes with weak interactions, maybe, your hydrogen bond, so forth. They can also be irreversible. If it's an irreversible inhibitor, it's going to be forming onto or grabbing onto that active site with a covalent bond. A lot of things that we think of as toxins and poisons are irreversible enzyme inhibitors. Enzymes evolved from other proteins back in the day. Some enzymes are very similar in many different organisms, from bacteria to us. So enzymes are coded for by DNA, and specifically genes, which are small subsets of DNA that code for proteins. Any change or mutation that leads to a change in the amino acid sequence of a composition will affect that enzyme. Altered enzymes might have new activity. We say novel activity because that's schmancy, I guess. But we do use that word novel a lot in biology. So it has novel activity. Maybe it can bind a different substrate, a slightly different substrate. And so that can cause the evolution of an organism. Under certain environmental conditions, that altered form might actually be favored over the original form. So enzymes can evolve as well, and that's part of the process of evolution of whole organisms. Interestingly, cells do highly regulate their metabolic pathways. There's a couple of ways that this happens. First of all, it can happen by switching genes on or off that compose or encode for specific enzymes. We're going to talk about this later in the semester. So for now, just think about a switch turning off a particular portion of the DNA. But one thing that we're going to talk about now is how enzymes are regulated once they're formed. And let's take a look at what that looks like. So one form of regulation is called allosteric regulation. And allosteric regulation can either inhibit or stimulate an enzyme's activity. Allosteric regulation happens when a regulatory molecule binds to a protein at one site and affects the protein's function at the other site. So let's take a look at this diagram and see what that means. Here is an enzyme, and this is, notice that it's got 4 active sites, 1, 2, 3, 4. And in the middle of this is something called the regulatory site. And there's actually 4 regulatory sites here too. 1, 2, 3, 4. So an allosteric activator, what that does is it stabilizes the enzyme in the active form. In other words, the enzyme's not going to do its job unless the activator goes into the activator site or the regulatory site. Going into the regulatory site allows the substrates to then bind to the active site, and you go forward and you have your active enzyme. Now, the allosteric enzyme with its 4 units, that can go back and forth between the active form and the inactive form. And with the inactive form, notice those active sites, they're not functional. They're the wrong shape. So what happens here is when it's in the inactive form, another molecule called an inhibitor comes along and it binds to this shape and it holds it in that shape, so that even if there's all of these substrates around here trying to bind onto this enzyme, this inhibitor is stabilizing the inactive form, and it's not going to do the job that you might think it should be doing. But this is all important. The activator and the inhibitor are both very important for making sure that your body's doing what it needs to do when it needs to do it. Most allosteric regulated enzymes are made from 2 or more polypeptide subunits. Remember, we talked about the quaternary structure of proteins. If you don't remember about the quaternary structure of proteins, you might want to go back and review that section of the notes. Enzyme complexes that are made up of all these 2 or more polypeptide subunits, they can oscillate back and forth between the active and inactive shape. And of course, we have an activator and an inhibitor. All of those are the terms that I'm wanting you to know for how allosteric activation and inhibition works. There's also this concept of cooperativity. Cooperate is the root there. And so what happens with cooperativity is that the binding of one substrate molecule to the active site of one subunit locks all the other subunits into the active shape. So there's our inactive form, and if a substrate can bind, then it basically holds all the other active forms and it's very stably open. And so having that substrate, it's cooperating, right? It's cooperative. It kind of amplifies and makes that enzyme do more of its job by binding to one of the active sites, holds it in place. There's also something called feedback inhibition. And I think feedback inhibition is really interesting because what happens with feedback inhibition is that the end product of a long metabolic pathway, once you get a lot of those end products being formed, those end products will bind to something called the allosteric site. That's not a term I used before, but if I were to go back and look at where we were talking about here, where that non-competitive inhibitor joins up, that's also called the allosteric site. So if we go back to here and we think about this feedback inhibition, such a cool concept. So let's start here with our enzyme, and we've got our active site and we have our allosteric site over here. And here's our initial substrate. I'm going to use the example. So this is threonine, and threonine is an amino acid. So this pathway, right? We talked about metabolic pathways at the beginning of chapter 6. This pathway is going to convert threonine into isoleucine. Isoleucine is also a kind of amino acid. So what we have here is threonine, it's in the active site, and that means it's going to go through all of these intermediates. We're not naming the intermediates right now, but it's got a series of enzymes involved in it. And eventually you make isoleucine after 5 enzymes have gotten involved and 5 intermediate molecules have gotten involved. But this isoleucine, this final product, it starts building up and building up and building up inside the cell. And what will happen is that isoleucine will bind to the allosteric site here, and it'll basically shut down by changing the active site. It'll shut down the ability of this enzyme to bind to threonine. What this is saying is, hey, body, we've got enough isoleucine. We don't need to convert any more threonine into isoleucine. Thank you very much for your job, but we don't need you to help us anymore. The last concept that we have for our enzymes and thinking about metabolism in general is that there are structures in the cell that help bring order and organization to metabolic pathways. If we go back to this metabolic pathway, we can see that we need the coordination of 5 enzymes. How do we manage that? Well, some enzymes are actually found in plasma membranes. In this case, we see an example of how the matrix and the membrane, the interior membrane of a mitochondria, which we're going to learn has just a lot of various chemical reactions that are happening, and they need to happen in a certain order. So we're going to see a lot of enzymes embedded in membranes. So specifically, when we get into the chapter on cellular respiration, we're going to be focusing a lot on the mitochondria, and I'm going to be showing you lots of pictures of enzymes embedded in membranes. Keep that in mind as we're going along and learning about our metabolic pathways in our specific examples of cellular respiration and photosynthesis. Well, I hope you've learned a lot about metabolism in this chapter, and if you have any questions, you know how to get in touch with me.